So I think you know that most of our genome is transcribed into RNA, and that the functions of most of these RNAs remains unknown. And also a number of diseases are due to defects in non-coding regulatory RNAs of many types, hence this meeting. So this is why we consider RNA as the new genetics. All right, so I don't know if you remember, it's almost 2.5 years ago now, there was a piece in the New York Times magazine about the Tell-tale RNA. And the idea behind this piece was that if we want a better diagnostic for doping in athletics, it's better not to test for the drug, but to test for the RNAs that were induced by the drug. And so in that spirit, I know that looks like DNA. Whoever the artist was doesn't appreciate that RNA is largely single-stranded, but you get the idea that RNA is the smoking gun, and that in the case of many diseases, or if we want to understand how our genomes are expressed, really it's better to accompany any DNA sequencing with an analysis of the transcriptome. So the first part of our meeting is going to pertain to long non-coding RNAs, and these are RNAs that are at least 200 nucleotides. Sometimes they've been called IncRNAs because some can be intergenic, and they are annotated to lack a long or a conserved open reading frame, translational open reading frame. Many, in fact, have the same structures as mRNAs, and they accumulate in the cytoplasm just like mRNAs. Generally, this type of long non-coding RNA derives from a pre-mRNA that has fewer splice junctions. In fact, some of them don't undergo splicing. They're generally expressed at lower levels than mRNAs. They exhibit weak sequence constraints and show limited phylogenetic conservation. And an example of this are the half SBS RNAs that my lab studies. These are ALU element-containing RNAs that some of which are not translated, but many, we know, turn out to be translated, but they're annotated not to be translated. And then there are other types of long non-coding RNAs that are highly abundant. They accumulate in the nucleus, and they have a 3' and triple helical structure generated by the tRNA biogenesis machinery. And examples of these types of long non-coding RNAs are the transforming mallet 1 and also the perispeckle component H-neat, which is also called MEN beta. There are the SNO long non-coding RNAs, the small nucleolar long non-coding RNAs. They also lack a 3' end poly A tail, and instead they have the SNO structures at their 5' and 3' ends. And consider the circular RNAs. They can be generated in high abundance by the pre-mRNA splicing machinery, and they are often promoted by double-stranded RNA binding proteins. These are circular and therefore resistant to exonucleolytic decay. All right, so this diagram shows where long non-coding RNAs can derive, and where they derive from influences their cellular function in many cases. So if you look at the gray regions, this is one protein coding gene, and here is another. And some long non-coding RNAs are divergent. In fact, it's thought that most of our protein coding genes have a bidirectional promoter where there's a promoter producing the pre-mRNA that encodes ultimately the message in the protein, and then a long non-coding RNA derives from the divergent promoter. You can see that some are intronic, some are intergenic, some can overlap in the same sense with a protein coding gene, and some are antisense. And to show you how complicated it can be, this is a portion of one of my lab's favorite regions of the genome. And there are transcripts that have been annotated and available to observe in the RefSeq and the Ensemble databases. And for a simplified version, you can look down here. So these are the two different isoforms that encode the protein. They're just part of the mRNAs. And there are long non-coding RNAs that overlap with that. And in the antisense direction, there are transcripts as well. And so this is a cautionary tale for anybody who's using RT-PCR to analyze their favorite mRNA, for example. Please look at the transcriptome databases to be sure that you're not also measuring something else, especially if you don't have a strand-specific way to make cDNA. So I highly recommend you look at the databases here to be sure you're measuring what you think you are, and not also something else. All right, so what are the outstanding questions that pertain to long non-coding RNAs? How should they be classified? Should they be classified by their genomic origin? So there are microRNAs that are close to transcription start sites versus those that aren't. I don't think that's the best way to go, but it's a possibility. Or should they be classified by function? And these are imprinting long non-coding RNAs. How many are functional? Which are confined to the nucleus, and how are they confined to the nucleus? How are they otherwise regulated? What is their abundance? Which are translated, and we know some of them are, and we know some of them are by their being, for example, targets for nonsense-mediated mRNA decay or Stauffen-mediated decay. We know that. Kristen Baker's lab has studied that as well. So even though they're annotated as not being translated, they very well may be. And of those translated, how many encode a functional peptide? And there are examples of that, and I'm sure there are going to be more coming in the future. So we're going to have 3 talks on long non-coding RNAs by Phil Sharp, Howard Chang, then after our break by Jeannie Lee. Then there are other types of non-coding RNAs. Introns can be considered a form of non-coding RNA, and of course microRNAs too. Ben Blenko will talk about splicing, alternative splicing, and Josh Mandel will talk about new mechanisms for non-coding RNAs and gene regulation. And then there will be talks on using non-coding RNA as a tool. And these talks pertain to the CRISPR-Cas9 system, which is a bacterial defense system. And it is a system that uses a non-coding RNA as a guide to guide the Cas9 nuclease, which can be used to induce site-specific DNA cleavage. And the damage is then repaired by cellular DNA repair mechanisms by either non-homologous end-joining DNA repair pathway, or the homology-directed repair pathway. And of course, the CRISPR-Cas9 system has now been harnessed as a programmable way to edit or modify genomes in mammalian cells in specific ways, and insertions and deletions, indels, can be used to generate knockouts of gene expression, and they often induce my favorite pathway, nonsense-mediated mRNA decay, or it can also be used as a means for introducing knock-ins. And I'm not going to go through the mechanism too much. This is just a simple illustration of how the pathway can be used using a single double-stranded cleavage and a single guide RNA, and of course more specificity will be achieved with more than one guide RNA nicking each strand in a specific way, and we're going to hear some talks on that. Oh, and I don't have the, yes I do. So what are the CRISPR-Cas9 questions? There are lots of them. What are the most effective CRISPR-Cas9 toolboxes? What are the ethical issues behind the usage to modify gene expression? And how can the ethical issues be overseen? And the people who will be talking about the CRISPR-Cas9 system, unfortunately, Emmanuel was not able to make a video, so we will not be having that presentation, but we have Feng Zhang, and also Eric Sonheimer is going to be talking about CRISPR-Cas9. All right, so that's my introduction. It gives you a taste of what is going to be coming next.